Our last speaker for this morning is Crystal Prather from MIT, talking about rational design of microbial chemical factories.
Yes, that's right. Switch me over. I have two challenges. One is that I'm the only thing standing between you and an opportunity to get up and stretch your legs and take in the beautiful scenery that was described. The other is that I'm still on East Coast time, and I was really hoping to get this done before midnight. So for me, it's pretty late. Let me start by thanking the organizers for the opportunity to be here, both to be in this place and to be in this company of people. My talk is going to be a bit of a departure from what you've heard earlier. We're still talking about synthetic constructions, but now we're talking about pathways. And I want to try to motivate it this way by saying that if we look historically, microorganisms have been wonderful examples of chemical factories. So more traditionally, we might think about those things that are considered natural products historically, that is antibiotics, antimicrobials, even therapeutics like lovastatin. This has moved on to lots of work for commercial scale synthesis of amino acids and organic acids. And traditionally, a lot of the efforts in this area has been on the improvement of natural producers. So as an example of how this might work, a cartoon, if you will, our objective here is to get our poor little rabbit to his favorite pet mouse, and he has a lot of different obstacles in his way. A metabolic engineering analogy for this would be that we want to be able to block off non-productive routes or competing routes. We may also want to stimulate our mouse to move a little bit faster with the ultimate goal of getting him to his end destination. Now, again, if we go back to the role of microbes as chemical factories, there are more recent examples where now we've gone beyond looking at natural producers and instead looking towards trying to reconstitute natural pathways into unnatural hosts. This really allows us to expand the synthetic capability of biology, okay, where if you think about it, biology is really a collection, especially a living organism, is a collection of a series of chemical reactions that are mediated by enzymes. Right? So our objective is to be able to maximize our ability to use those enzymes. And I want to argue that microbes, as I said, have been very effective as chemical factories, and that there will be a continued and perhaps even an increasing need to use microbes in this way or to use biology in this way. This is a diagram that shows for the U.S. I don't have the numbers for global oil supply, but a barrel of oil is different in different parts of the world, and the distribution in terms of what happens to it is different in different parts of the world. But this is taken from a report last year from the Department of Energy in the United States showing what happens to a barrel of crude oil. So where does it go? And this is the area that we're used to thinking about. Roughly 2/3 of it goes into gasoline or diesel fuel, the things that are powering the automobiles that are all over our highways, making it difficult to get to and fro. And you'll hear talks later on today, as well as a whole session tomorrow, dedicated towards production of biofuels. And the talk before mine also looked at one aspect of that, which is being able to engineer the feedstocks in this manner. I want to point out, though, what's the next largest slice of the pie. And this is labeled here as other products, as if these are things that don't really matter. But these are, in most cases, chemicals. Okay? These are the things that start as the building blocks that go into all sorts of materials, including most of the plastics in the world, materials that go into our laptops, et cetera. And so while there may be arguments that ultimately you want to go towards a hydrogen economy, and I'm not arguing either way, but there may be arguments for hydrogen economy for transportation or fuel cells for transportation, this is talking about carbon now, right? Things that can't really be supplanted if you want to look at what happens to petroleum feedstocks. And so in this increasing desire for microbial chemical factories that are going to make now different molecules that we might be used to, this maize analogy may no longer work. Okay? So it may be that we can't really look at an optimal route for a maize. Our mouse has to get a little bit more creative, and maybe we're just going to demolish the maize and say we're going to get there faster. Okay? I have very creative graduate students, and I steal their slides. Don't tell them they're not here. So we can look for inspiration in terms of a route to do this by looking at the field of biocatalysis, which has had a lot of success in looking at the utility of enzymes to do chemical reactions. And the key here is to rely on a functional specificity across a broad substrate range. And here's an example of that. One enzyme, phenylalanine dehydrogenase, whose natural function is to convert the ketoacid precursor of phenylalanine to the L-amino acid. And this enzyme was used successfully in an example given here from Bristol Myers Squibb, from a pharmaceutical company, to convert now this precursor into its homologous amino acid. Okay? And the take-home message here is that if we can find enzymes capable of converting unnatural substrates, or not just find them, engineer enzymes that are capable of doing this, then we open up the door to be able to make more and more unnatural products. Okay? And if you start off in an introductory level biochemistry class, you might think there's no way this enzyme will work on this molecule. But again, the business end of it is the same. It's a keto acid here, a keto acid there. And I'm not arguing that the activity is the same, that the kinetic parameters don't change, but this was sufficient in order to produce material that ultimately went into making quantities large enough for clinical trials. So with that as a backdrop, what we try to do is something that we call retrobiosynthetic pathway design. The basic idea here is to say, okay, we want to make molecules, and we don't necessarily know what that maze is going to look like, so we're going to be looking for little pieces to put into our explosives. We have biocatalysis, which I explained, and also bioprospecting. And this is what I'm really going to talk about today, and that is being able to search around nature for reactions that we're interested in and put them together in new ways to make pathways. Okay? This is going to guide our part selection. We have a rich history in metabolic engineering that's very complementary to synthetic biology that gives us tools for both assembling our systems, these collection of parts, into higher order systems, and more importantly, being able to analyze those systems, understand where the bottlenecks are, and how to make them better. Our goal here now is to take this theory behind biocatalysis and try to design and assemble now an in vivo series of conversions where we ultimately want to be able to tie our starting reactants back to something that the cell already makes or that it readily consumes. Now, at this point, I want to say that there are existing biosynthetic pathway algorithms. In particular, I'll highlight the work of Vasily Hatzimanakadis and Linda Broadbelt. Vasily used to be at Northwestern, where Linda is and is now at EPFL in Lausanne, Switzerland, who've spent quite a bit of time looking at computational methods for designing pathways and different considerations for doing that. Larry Wacken and Linda Ellis at the University of Minnesota have had an effort for quite a while to make a biocatalysis and biodegradation database that helps to look at designing pathways as well. And then Alfonso Jaramillo, who's here at this meeting, will give a talk tomorrow. He and I did some work together looking at ways to get to pathways as well, and this work will be published very soon in bioinformatics. I encourage you to go to his talk and hear more about that algorithm. I am fundamentally an experimentalist, however, and so the approach that I take is an experimental approach where we go through, the way I describe it to prospective graduate students is we pick molecules, we design pathways, we go try to make them. And we have problems. Lots and lots and lots and lots and lots and lots of problems. Right? I explain to people, that's why they call it research, because you keep doing it over and over again. And as we try to solve those problems, we're trying to elucidate some design principles about the best ways to go about this process in the future, and as well, try to develop some design and assembly tools, so some specific devices. Unfortunately, I'm not going to have a time today to talk to you about some of our work in this area, and I really want to just focus on giving you an example of pulling enzymes together and seeing what happens in that case. So here's our target compound. I won't read all of this. The take-home message to those words is that we picked a compound that matters. And in particular, it's been identified as a value-added chemical from biomass with the study. These are two of our national labs in the United States. The other take-home message here is that there's no known microbial pathway for this. And so if we say, well, one of the points before was that this is something that's found in fruits and vegetables and mammals, and there is a pathway now, the mammalian pathway, either from galactose or from glucose, and eventually you get to glucaric acid, and this is where the mouse is like, yeah, I'm not really trying to go there. So we tried to look for dynamite, okay, and we did this in a bioprospecting approach, and I'll explain to you more of an engineered approach or design approach towards the end of the talk, where we looked at the literature and said, okay, if I look for glucaric acid because it's a natural product, where does it show up? And it turns out you can get glucaric acid from a compound called glucuronic acid using an enzyme from Pseudomonas. And if you say, well, how do I get this? You can get that from myoinositol using a compound, or an enzyme rather, called myoinositol oxygenase. This we got from mouse. There are other sources of it. And you can get myoinositol from glucose, and more specifically from glucose 6-phosphate, using one cloned enzyme, but it actually represents 3 different steps in E. coli. Namely, the transport of glucose into the cell through the PTS system, which gives you glucose 6-phosphate. Our cloned enzyme does our isomerization reaction, and then there's an endogenous phosphatase that will cleave the phosphate off and give us free myoinositol. This one we cloned. There was, again, a reference that showed us that would work. This one we had to synthesize for codon optimization in E. coli. And I want to spend just a few minutes talking about this enzyme because this is an example. My abstract promised to tell you about some of the challenges. This is an example of a challenge where a couple years ago when I talked about the kinds of things we're interested in, people would say, well, you know, if you find some reference to it in the literature, then there's got to be a sequence available for you, and you just call Blue Heron or GeneArt or DNA 2.0, et cetera, and you'll get it in a couple of days. We found references to this from the 1970s. Not doing gene sequencing then. Didn't have a protein sequence there. We had a reference, two references, one published in the 60s, one published in the 70s, that said this enzyme, or it said the organism contained an enzyme that was capable of doing this reaction. And so it actually took us a little while to figure out how to get this enzyme very easily and realize the following, and that is that E. coli, wild type especially, is capable of consuming both glucuronic acid and glucaric acid for growth, but the pathways are independent of one another. And the enzyme that we're looking for converts glucuronic acid to glucaric acid. So if we start with a mutant that's incapable of growth on glucuronic acid, it will grow, this is just on glucose or complex medium, on glucaric acid. And then if we use that as the host to screen our genomic DNA library, then we may pull out the enzyme that's capable of converting glucuronic acid to glucaric acid, and then we'll get growth of those growth of the cells under those conditions. And that's exactly what we did, and pulled out a couple of candidate molecules, one of which we still don't know exactly what it does, but it gave us a signal in terms of being able to make NADH, and the second of which was very promising because it had a reaction rate that was proportional to glucuronic acid concentration. And we knew from our reaction here that we had an oxidation reaction, which meant that we were either oxidizing this aldehyde or oxidizing one of these alcohols. And by mass spec, we were able to confirm that we did indeed add an oxygen, and our sample purified from the enzyme reaction matched up very well from our standard, which means we got the enzyme that we were looking for. So then we put all these three together. And again, the first two have been described, so I won't talk in much detail about them, and got the following results, which is that we made a decent amount of material, about a gram per liter. I've had some people say a gram per liter, that'll never get you anywhere. And the folks who work with plant metabolites go, a gram per liter, how fantastic. So, you know, it depends on who you want to be your friend that day. And what was interesting in this case is that this was a three-step pathway again. We were accumulating myo-inositol, not accumulating glucuronic acid at all, which meant that what we were looking at was a system where myox was the rate-limiting enzyme. This is the second step on our pathway. And when we looked at the enzyme activities, that was confirmed. This third enzyme, which we were the first to clone, has wonderful activity, and you can see the other ones are not getting anywhere close. And in particular, our myox gene was causing some problems. And looking more closely at this enzyme revealed a few things. This is now looking at that enzyme by itself, so not in the three-step pathway, but just seeing how it expresses from the synthesized gene, the optimized gene, I should say. This is now activity in nanomole per minute per milligram, so the same scale as on the previous slide, and I'll come back to that data in just a second. And there were two things to notice about this. One is that this enzyme appears to be intrinsically unstable, okay? Meaning, if we looked at our activity at 6 hours, so this was early on in the mid-exponential phase, it was relatively high. If we looked at that same activity at 24 hours, so a day after induction, it was relatively low. And even though it had dropped off considerably, we still were able to get product form if we fed myo-inositol to this system, okay? Now, by comparison, the numbers on the previous slide were around 10. Okay, so this means that we're not getting anywhere close to the maximum capacity of this enzyme. The other thing to keep in mind, though, or to notice rather from this slide, is that we also saw this very strong dependence on having myo-inositol present. And this was something that had been referred to in the literature that we observed as well. And this is not, mind you, a reaction rate that depends on the substrate concentration in the in vitro assay. This is if I grow the cells, E. coli cells now, in the presence of myo-inositol versus in the absence of myo-inositol, wash away the medium, so now all I have is cells, break those cells open, and do in vitro assays with the same concentration, you get more than a 10-fold difference in the activity between these two. Okay? This told us, as far as what are the challenges we're going to face with this pathway, that we need to have high myo-inositol production by this first enzyme in order to get maximum activity from our system. The other thing it tells me from a synthetic biology perspective is that this is not a very well characterized part. Nobody really understands what it is about having the substrate present in this system that contributes to higher activity. Does it assist with folding? Does it in some way activate the enzyme in a post-translational way? No one is really clear. So this is one of the struggles where if we're looking at enzymes and trying to pull them out, trying to assemble them together in predictable ways, the lack of some fundamental biochemical information can make that difficult. It doesn't mean, however, that we can't still try to come up with creative solutions to that problem. And so this is work that was done in collaboration with John Duber, who's part of the Synthetic Biology Engineering Research Center. And the idea here is that if we look at expressing these two enzymes independently, INO1 is going to make myo-inositol. It's going to have some distribution of concentration distribution in the cell just based on the diffusion of that molecule through the cytoplasm. If instead I use a synthetic protein scaffold that has tags that allow me to now co-localize my two enzymes, the theory, or at least what we wanted to test, was whether or not this would increase the local concentration of myo-inositol and therefore increase the concentration that this enzyme sees, leading to better activation, whatever that means, because we still don't understand the role that this plays, and hopefully better conversion in our system. And so this is the result, first of all, just looking at the first two steps of this pathway, since the second enzyme was the one that was really a problem. And you can see that we do get an increase in the amount of that compound that's formed, glucuronic acid, having the scaffold versus not having the scaffold. But the real question was whether or not this gave us better activation of myox. And here, it depends on how much you trust error bars, right? Maybe the error bars are wrong and the averages are correct. You see an offset here, but again, if we look at the error bars, we're not really sure if the effect here is one that we were aiming for, which is to increase the activity of our myox by increasing the local concentration. If we put all three enzymes together, we actually had to do a little bit more optimization around the inducer concentrations. And in this case now, you also see with the scaffold now a much larger increase in the amount of glucaric acid that we get with the scaffold relative to without the scaffold, now again after having optimized it a little bit. But once again, when we look at the differences between the myox activity, the second enzyme, we see a little bit of a difference, but not necessarily the dramatic improvement that we were hoping for. Okay? So I still have this as a question mark because those error bars are so close together that it's hard to really believe it. One of the other challenges we have, however, is that the assay for this enzyme is a pretty poor assay, and so we're hoping that by improving the assay, we can shrink those error bars and have more reliable data. So let me give you a little bit of a summary of what we've done so far. The initial construct gave less than half a gram per liter of glucaric acid. There was some optimization of culture conditions. I didn't point out that the myox gene requires molecular oxygen as a substrate. And so this was all done in shake flasks. The agitation rate of those culture flasks actually made a difference in terms of being able to supply sufficient amounts of oxygen to get that enzyme to work well. And then there was this bit that my student who does this calls parts optimization, where we looked at things like copy number, promoter strength, et cetera. And I want to spend just a minute talking about this as a challenge because I do think it points out one of the ways in which, as a synthetic biology community, we have to try to understand how these systems behave to make them more predictable. Okay, so this is glucuronic acid, the same slide that I showed with the scaffolding before, and this is now compared to a system where we use a T7 promoter. Okay, this doesn't look so great. All right, and people say, well, I get that because if you read through the literature, you have a rich history that talks about metabolic burden and says what happens when you go too far the other way is that you make too much of the enzyme, it doesn't really follow property, and so if you look at a curve of what your activity is as a function of your either gene copy number or increasing promoter strength, you usually get an optimum and then it drops off. The problem with that theory in this case is that when we actually looked at enzyme activity, the enzyme activity where we had a T7 promoter was actually the highest activity, but the productivity of the system was the lowest. And the same thing was true if we looked at the three gene system making glucaric acid. The effect wasn't quite as bad. We still got more glucaric acid here than we had gotten of glucuronic acid, primarily because that third enzyme is so strong that it pulls flux through the pathway. But again, if we look at the activities here, we're not seeing a large drop-off in the activity of the system. So what our theory is, and we looked at now glucose utilization, and what we believe is happening in this case is that when promoter is too strong, what we actually observed was a decrease in glucose uptake rate. And so the whole system ends up being downregulated. We get lower biomass yield, and we also get lower productivity in this case, okay? Which, again, okay, we can explain it. The challenge I have from a synthetic biology perspective is that I think we need better tools to be able to have predictive modeling of the metabolic burden effect. Okay? It's very easy, I'll put in quotations marks. My students wouldn't agree with this. But after the fact, you can use hindsight to look at your system, to see what happened, to try to do some diagnostic assays, and be able to say, okay, I know what happened, I can do better the next time. But to really be predictive and to have the kind of design strategy that we want for synthetic biology, I think this is an area where we're going to have to really try to do some work to understand how these systems behave. It's also an area where, for the first speaker in this section, for Clyde Hutchison, if we have simplified organisms, he talked about being able to better understand how metabolic pathways are incorporated into those, and I think that's a very promising area of research. I'll just end by saying here, by being able to pull in these scaffolds as well, we're able to get up to nearly 2 grams per liter, starting from an initial concentration of about 1/4 of a gram per liter. Just very quickly, I want to talk about, and I'm not going to go through all this, this is just a slide that's intended to show that we did also look at designing routes using more of this biocatalysis methodology, where we looked generally at the capability of enzymes to perform chemical reactions. And in particular, we came up with the following 3-step pathway, where the last step is the same, so that's good, because that's our best enzyme from our bioprospecting strategy. So we want to be able to pull out the one that works the best there. But for these 2 enzymes, they're both cases where the natural enzyme does this reaction. So there's a galactose oxidase enzyme that oxidizes this alcohol to an aldehyde, but it doesn't naturally accept glucose as a substrate. So we're working with Codon Devices to re-engineer that enzyme, and we're in the process of screening these libraries to look for something that has reasonable activity on glucose. And then the second step now is to take this aldehyde and convert that to a carboxylic acid. This form of glucuronic acid exists in equilibrium with the linear form that I showed you before. Okay, so this is the same molecule, just like glucose has a hemiacetyl form versus a linear form. This is a collaboration that we're doing with Alfonso Jaramillo, who I mentioned previously, where there's a natural enzyme which accepts, in this case now, benzaldehyde, as well as a number of different substrates, so that we still have more of a ring or planar structure, and then an aldehyde group from there. And again, Alfonso is using his computational software in order to look at ways of redesigning that enzyme. And Codon Devices, I'll point out, is also a sponsor of this meeting, so we have folks from both of these companies here today. I'll just end now with acknowledgments, particularly by recognizing Tae-Sok Moon, who is the graduate student, and Sang-Wol Yoon, who's the postdoc, who did most of the glucuric acid work. I want to acknowledge our funding sources, in particular the National Science Foundation through SINBERT, our collaborators who I've mentioned throughout the talk, Alfonso from Ecole Polytechnique and John Duber from QB3 at Berkeley. Again, QB3 is a sponsor of this meeting. I just want to end by acknowledging the ERC here, not for funding, but for really the rich intellectual environment that being a part of this center has created for me and for my students and postdocs, for the opportunity to talk about a lot of these challenges on a regular basis and be really creative in thinking about our solutions. And with that, I thank you for your attention. I'm happy to take questions. Everybody's really hungry. Yes.
Our expression vector permit to control gene expression either by copy number or by promoter activity or both. And it's a little difference which way. So what do you find out?
Yeah.
It's better copy control first and then gene expression, or first gene expression and then control of copy.
So the question is, you can control gene expression either by looking at copy number or by looking at promoter, and which should you do first, right? Or which?
What's your experience?
So my experience is that it's a case-by-case basis. In this particular case, we actually looked at lower copy number plasmids first, and we got terrible expression. And so we had to go to high copy plasmids. And once we were at high copy plasmids, then we could play around with the promoters. But when we played around with those promoters at low copy number,
and how do you increase the copy number?
In this case, we increased the copy number by using plasmids with different replicons.
Oh no, you should control copy number by using TRFA and OREV, some origin which could be induced.
Yeah, but so OREV is going to give you a very low copy number, right? Copy number of 1, and then you can't amplify it.
TRFA and it goes high.
Right. So, but it depends on what your objective is, right? So if you do that, then what you're doing is waiting for amplification of your gene, your gene dosage rather, before you get now amplification of your protein. So it's going to take time to propagate through that system. Okay? And it also requires you to have an additional level of control. So now we have to try to control plasma copy number while still controlling expression. I'm not saying it doesn't work. I'm saying it adds a level of complexity to the system that we weren't prepared to deal with at this time. Right? When we did look, though, at using a plasma which had a naturally lower copy number versus one that had a higher one, in this case we found that we couldn't get sufficient expression, although we had activity. So we had enzyme activity, but we couldn't measure product using the lower gene dosages, and so we ended up with higher copy number plasmids. In my previous life, I wrote papers about how low copy plasmids were great and they were going to be the answer to all my problems, and then they turn out not to be. My students like to remind me of that.
Okay.
I think there's
Your talk was a great example of how if you want biological end products, you've got a huge amount of biochemistry and organic
Yeah.
chemistry to go and get it.
yeah.
What will be the answer for your problem? Do you see a lot of people...
I knew that. I'm sorry.
A lot of people predict that the falling cost of gene synthesis is
Yeah.
going to revolutionize synthetic biology.
Yes.
Do you think that in your example that's the case? And if not, what do you think will revolutionize what you're doing?
So the falling cost of gene synthesis certainly is a very important role to play. Okay, and as an example of that, one of the things that it allows you to do, in the case, I'll look at the collaboration that we have with Professor Jaramillo. Okay, what he's doing is modeling that enzyme and proposing for us, here are variants you should make of that enzyme. The falling cost of gene synthesis makes that next step much, much simpler than it used to be. Okay? It gives us access to the diversity. Being able to rapidly synthesize DNA doesn't answer questions about biochemistry. It gets you faster towards being able to do the analysis and do the experiments that will give you the biochemical answers. So, I mean, I think this, to me, is one of the challenges with this particular area of applications of synthetic biology is really taking full advantage of the tools, but trying to understand to what extent we can have, for example, an abstraction hierarchy around biosynthesis, right? Given the fact that there are details like the fact that this one enzyme needs a substrate, but we don't really know what role it plays, you know, how thickly can you draw those lines? And I think that's an open question, but certainly the foundational technologies and the fundamental tools will speed up our process of discovery in tremendous ways. One more question?
Hi. I was curious about the increase in flux you saw when you added the scaffolding. Interesting because it's surprising that we can make the improvements because you would think evolution would have done that anyway. Why do you think nature hasn't done that but we can?
So in this particular case, these were enzymes that were never designed to act together. Right? One comes from Saccharomyces, one comes from mouse, one comes from Pseudomonas. I got a yeast, a mammal, and a bacterium. That sounds like the beginning of a joke. What happens when they walk into a bar, right? They walk into a bar and out comes glucaric acid, right? So in this case, with enzymes that weren't ever designed to work together, then I think very clearly that's what's happening there. But I will say, even with enzymes that may be designed to work together in a pathway, John has applied this technology to the valinate pathway with Jay Kiesling. And those are enzymes that do work together. But nature can go even further if you look at polyketide synthesis, for example, of actually physically linking these molecules together. So nature is optimized for its particular environmental conditions, but that's not necessarily optimized to produce goo-gobs of this thing that we want to make that nature didn't know we were interested in.
Okay. If you have any further questions, you can always ask the speaker.